Good afternoon and welcome to today's lecture. The lecture for today serves to be really just a review of topics that you've covered in previous classes. If the topics that we are covering today are new or unfamiliar to you, you should really spend some time going back over today's lecture and understanding the concepts and principles of what I'm going to cover in this lecture. So again, this is meant to be a review. It really isn't any new information, but if you are having difficulty with any of the material that I'm covering today, please feel free to either email me or just continue to review the material so that you have a really nice, strong foundation to continue on in our subsequent lectures. The first review topic that I want to talk about really briefly is that there are in fact two different cell types or categories that exist on Earth. The main categories of cell types are referred to as either prokaryote, like you see in the slide here, or eukaryotic or eukaryote, as you'll see in the subsequent slide. A typical prokaryotic cell is a cell that is defined by the fact that it doesn't have a specialized structure known as a nucleus that contains the DNA or genetic material. Now, prokaryotic cells do in fact have DNA, they just don't have a specialized structure known as the nucleus to house that genetic material. Prokaryotic cells tend to be things that you may typically think of as bacteria or archaea overall. You can contrast the prokaryote or prokaryotic cell to that of what you see on this particular slide, known as a eukaryote or eukaryotic cell. Eukaryotic cells can typically either be considered animals or plants. There are other categories of eukaryotic cells, but for most purposes and in most introductory courses, we focus on animal cells and plant cells. Eukaryotic cells are a specialized type of cell that has organelles, which are structures that have specialized function within the cell. The most important distinguishing feature of a eukaryotic cell is the fact that it has a nucleus, which is a specialized structure, an organelle, that houses the DNA or the genetic material for the cell. This particular slide is really just a nice summary that compares both prokaryotic cells to eukaryotic cells. Again, the main differing characteristic is that prokaryotic cells do not have a nucleus, a specialized structure for the DNA, and eukaryotic cells do in fact have a specialized structure known as the nucleus that houses the genetic material. Typically, prokaryotic cells tend to be relatively smaller than eukaryotic cells. Their genome or genetic material is usually contained in one circular DNA molecule, whereas in eukaryotic cells, there typically are multiple linear DNA molecules that make up the genome. The DNA itself in prokaryotic cells is not complexed with proteins known as histones, whereas in eukaryotic cells, the DNA is in fact complexed with proteins known as histones. If you continue on to take a molecular genetics course, you will learn a lot more about the differences in DNA, chromosome structure, and that type of thing when we compare prokaryotic and eukaryotic cells. But for our purposes, it's just really important to understand those basic differences that are highlighted on this slide. In comparison, prokaryotic cells typically have much less DNA overall, that their genome size is much smaller than a typical eukaryotic cell. And then lastly, another defining characteristic is that prokaryotic cells do not have specialized membrane-bound organelles, whereas eukaryotic cells do. So why review these basic types of cells overall at the beginning of a genetics course? The reasoning for that is our first topic that we're going to jump into is transmission genetics. That is how traits get passed on from one generation to the next. And in those examples, we are dealing specifically with eukaryotic organisms, eukaryotic cells. However, when we jump into molecular genetics towards the last third or last quarter of the semester, we will be talking about both prokaryotic examples of genetic mechanisms as well as eukaryotic mechanisms. And so again, it's really just important to make sure that you understand the differences between these two cell types before we move on. So the next concept that I want to touch on that hopefully should be a review for you is the concept of what's referred to as a homologous pair. So first what we'll do is we'll take a few steps back and define a few terms, and then we will continue on with the concept of homologous pairs. The first two definitions that are going to be repeated throughout the semester are definitions that deal with the amount of genetic information in a given cell. The term haploid refers to a cell that carries one complete set of genetic information. That's differentiated from the term diploid, which refers to a cell that carries two complete sets of genetic information. Most prokaryotic cells are found in the haploid state, meaning they have one complete set of genetic information, and most eukaryotic cells are found in the diploid state, meaning they contain two complete sets of genetic information. Now, obviously, there is some flexibility with respect to organisms and diploid and haploid state, so those statements I just made are generalized blanket statements overall. In addition to the terms haploid and diploid, I want to add the term chromosome. Now, a chromosome is defined as a structure that houses the genetic material and is typically made up of both proteins and DNA. DNA, of course, is the genetic material, but chromosomes are the specialized structure that the DNA is complexed in overall. When we talk about chromosomes in a typical diploid organism, that's where the term homologous pair comes into play. Because diploid organisms have two complete sets of genetic information, it also means they have two copies of every single chromosome present in those cells. Homologous pairs represent a pair of chromosomes that are identical with respect to the genes that are located on them. Homologous pairs of chromosomes are roughly the same size and the same shape as one another, but the most important thing that distinguishes a homologous pair is that they have the same genes located on them. The way that individuals get a homologous pair in a typical diploid organism is that one complete set of genetic information comes from the mother and one complete set of genetic information comes from the father. In humans, for example, there are 23 pairs of chromosomes in a diploid cell. 23 chromosomes came from the individual's mother, and 23 chromosomes came from the individual's father, for a total of 46 chromosomes in a typical human diploid cell. The homologous pair, again, is going to be where one copy came from the mother and one copy came from the father. Now, the majority of the rest of this review lecture is going to focus on nuclear division, specifically mechanisms of mitosis and meiosis, because they are at the foundation in our understanding of typical Mendelian transmission genetics. But in order to do that, we need to, again, set some standard for our understanding of chromosome structure, because chromosomes, again, are these specialized structures that house the genetic material. And in order to continue on with both mitosis and meiosis, we have to understand some fundamentals about these structures. Typically, when we see a picture of a chromosome, it looks very much like what you see in the center of the slide with the X-like structure. However, unfortunately, that tends to be a little bit misleading, in that a chromosome is not typically found in this characteristic X-like structure. In both of the figures, what you can see is a single chromosome. So in the left-hand sided figure at the very top, you have a structure that represents a single chromosome. What happens during the preparation for mitosis or meiosis is that all of the genetic material is copied or replicated or duplicated. So in the center of that figure on the left-hand side of the slide, you see that characteristic X-like structure. This represents a duplicated chromosome. The chromosome at the top represents the original chromosome. After replication has occurred, you get that characteristic X-like structure where the copy of the chromosome is physically attached to the original chromosome structure. In that same figure on the left, you have an electron micrograph image of a chromosome so that you can actually see what the chromosome looks like in a duplicated state. It has that classic characteristic X-like structure. When cell division occurs, whether it's mitosis or meiosis, there is a point where those two copies have to separate from one another, and that's what you see at the bottom of that figure, is the two structures physically separating from one another. What I also want to identify at this particular point on this slide is what holds those copied chromosomes together. And that is a specialized structure where there is a condensed portion of the chromosome known as the centromere, and more specifically, a portion of the centromere is known as the kinetochore, and that is essential for the processes of both mitosis and meiosis. So in the right-hand side, you have a more cartoon version again of a single chromosome. Then in the center of that figure, the copied or duplicated chromosome where the two copies are physically attached at the centromere. And then honing in to show you a portion of the centromere known as the kinetochore. Now, in the intro lecture that you listened to prior to this particular lecture, I mentioned at the tail end of that lecture that genetics is actually a very broad field and that there are subdivisions or subcategories of genetics that we will focus on throughout the entire semester. One aspect that I mentioned is something referred to as cytogenetics, which is really the study of the chromosomes themselves. So in this particular slide, what I want to do is highlight a little bit of information about the structure of a given chromosome. So if you focus your attention to the figure in the center, this is what is referred to as a karyotype, which really it is the first stages of a karyotype. We'll talk about how karyotypes are performed in some lectures later on down the road, but quickly or briefly, what happens in the preparation of a karyotype is chromosomes are isolated, they are stained, there's a picture taken of them, and then they are rearranged in order of chromosome number. And this is a way for scientists to then study those chromosomes. But one thing that has been described is the location of the centromere. So the centromere, like I mentioned in the previous slide, is the specialized structure that is a more condensed portion of the chromosome, and it is where two chromosomes that have been duplicated are attached to one another. Those two copies of a given chromosome are referred to as sister chromatids. So the X-like structure that you typically see, again, with a chromosome really represents a duplicated chromosome with the original chromosome and its duplicate physically attached at the centromere. At that point, each one of the chromosomes are referred to as sister chromatids. But again, for this particular slide, what I want to do is highlight that there are some structural differences in chromosomes. And this is dictated by where the centromere is located. So if you focus on the second structure down, you'll see the term metacentric. And I'm going to start here because this makes the most sense. When you have a centromere that is smack dab in the middle of the chromosome structure, where you have roughly equal amounts of chromosomes on either side of the centromere, this is referred to as a metacentric chromosome. It means that the centromere is smack dab in the middle. Now, if you look at the figure just above that, that's labeled submetacentric. This refers to a chromosome where the centromere is shifted from the center just a bit. And so you have portions of the chromosome that are a little bit smaller with respect to where the centromere is, and then portions of the chromosome that are longer. If you go all the way down to the bottom of the figure, you see the term acrocentric, and this refers to a centromere that's really close to the end of the chromosome. So there you see very small portions of chromosome on one side of the centromere and very large or longer portions of the chromosome on the other side. Lastly, you can see the structure with the term telocentric associated with it. This refers to chromosomes where the centromere is completely at the end of the structure. So these are the four broad categories that you can typically find chromosomes in with respect to the location of their centromere. This again should be new material. It shouldn't be a review. I'd be very surprised if you've heard all of this terminology in a previous course. So this right here is a little bit of new material. So what I want to do now is throw out a quick concept check to make sure that you've got some concept of things that we've talked about in this lecture up until this point. So in this case, the question asks, diploid cells have A, 2 chromosomes, B, 2 sets of chromosomes, C, 1 set of chromosomes, and D, 2 pairs of homologous chromosomes. So at this point, I'll let you go ahead and pause the lecture, think about that question, and choose your answer before you continue on with the lecture. Okay, so now that you've transitioned to this next slide, you can see that the answer is B, 2 sets of chromosomes. So when we talk about diploid cells, they are cells that have 2 complete sets of chromosomes in them. That's different from a typical haploid cell that has 1 complete set of chromosomes in it. Now, every living organism has a substance that we refer to as the genetic material, and this genetic material in most cases is in fact deoxyribonucleic acid. The DNA, or deoxyribonucleic acid, is organized into chromosomes, and we've highlighted a little bit of that in the beginning part of this lecture. In prokaryotic cells, there's typically 1 circularized chromosome, and in eukaryotic cells, there can be multiple linearized chromosomes. Regardless of the chromosome structure, DNA needs to get replicated or copied when new cells are being formed. And this must be an extremely precise mechanism so that every new cell gets an entire, complete set of genetic material required for that cell to continue to survive. So what we're going to continue on with in this review is discussing the 2 major types of nuclear division, referred to as mitosis and meiosis. In eukaryotes, mitosis is a form of nuclear division that results in the production of 2 nuclei having the same chromosome complement as the original nucleus in question. When we add the term cell division, this is when the cell will result in 2 cells at the end of the process. So mitosis is really a nuclear division mechanism. When that is followed by a process known as cytokinesis, we then have complete cell division. So the terms cell division and mitosis are not entirely synonymous. Mitosis refers to nuclear division, cell division refers to the actual cell dividing. In addition to that, we have the term meiosis. Now, meiosis is a process in which chromosomes of eukaryotic cells divide and give rise to haploid nuclei. So in meiosis, you start off with a diploid nucleus, and throughout the process, the end result is giving rise to haploid nuclei. Starting off with a nucleus that has 2 complete sets of genetic information, and nuclei then that result that have 1 complete set of genetic information. Meiosis is a process that is necessary for sexual reproduction to occur, and again, we're going to review those mechanisms in this lecture. Now, in previous courses, you may have heard the term cell cycle. The cell cycle is really just the life cycle of a given eukaryotic cell, which is what we're typically referring to, from the time of it arises from a division to the time that that cell actually divides itself. What you see in this figure is a representation of that mitotic cell cycle. So at the top of the cycle at 12 o'clock, you see where a cell division has just occurred, giving rise to 2 cells. The cell cycle then begins at that particular point where that cell has arisen from a cell division event. What you see for the majority of the time of the cell cycle or the life cycle of the cell is that the cell is in a stage known as interphase. Interphase, represented by the lighter green color of the cycle on this figure, is a time for the normal everyday life of the cell. A very small portion, designated in the reddish or pinkish color, represents mitosis, where the cell is going to continue on with nuclear division and eventually lead to the production of 2 new cells in a cell division cycle. So interphase is generally referred to, again, as the stage where the normal everyday life of the cell. It is subdivided into 3 distinct categories or subphases. There is the G1 phase, which stands for gap 1 or first gap. There is the S phase, which is short for synthesis, and there is the G2 phase, which is the second gap. In the first gap, or G1, the cell begins to grow and get larger because, again, it is from the time point of when that cell arose from a cell division event. In the S phase, the genetic material is copied or replicated or synthesized in preparation for a mitotic event. So the S stands for synthesis, specifically synthesis of DNA. And then G2 is just another continued growth and everyday life component of the cell in question. And then after the G2 is when the cell will enter into mitosis, which is the nuclear division phase, and if it is followed by cytokinesis, results in cell division. So mitosis, again, is defined as a mechanism of nuclear division. It is when the chromosomes or the genetic material is properly separated so that they can eventually result into separate cells. So when we talk about mitosis, the focus is always on that genetic material and what's happening to the genetic material during each one of those stages. On the far left, you can see late interphase. Late interphase means it's just ready to enter into mitosis. That means that the S phase of interphase has taken place. All of the genetic material has been duplicated. However, if you looked under the light microscope during interphase, you wouldn't actually see any distinct chromosome structures. It's because they are in a much looser orientation during interphase, but given that this is late interphase, all of the genetic material has been replicated. The first stage of mitosis is prophase. In prophase, the chromosomes begin to condense, and the nuclear envelope or the nuclear membrane begins to break down. We also see organization of cytoskeletal components known as mitotic spindle or microtubules. They begin to organize, and you can see them in this structure or this figure. In prometaphase, the last figure on this slide to the far right, you can see that the chromosomes have completely condensed. You can see that the nuclear envelope is pretty much all broken down. And you can also see that those microtubules have attached themselves to the centromeres of the chromosomes. Specifically, those microtubules attach to the kinetochores of the centromeres. And this is, again, very characteristic of prometaphase. The next stage is metaphase. And in metaphase, the chromosomes align along a central axis or central plane of the cell. The way that the chromosomes do this is because the microtubules have attached themselves to the centromeres, and that pushes and pulls the chromosomes so that they are in this location along the central axis of the cell. And that is a clear defining characteristic of metaphase. In anaphase, what you begin to see is that those duplicated chromosomes start to pull apart from one another. The microtubules that are attached to the centromeres begin to pull those chromosomes apart. So those two copies that were physically attached to one another are now pulled apart in opposite directions of the cell. And that is a clear distinguishing feature of anaphase. In telophase, you begin to see the chromosomes decondense or uncoil. You begin to see that nuclear envelope reforming, and you begin to see what will be or what will result in the dividing point of the two cells. Now, that represents mitosis, which is the nuclear division process. Cytokinesis, like I have labeled at the very end of this slide, is the final step of cell division. That is where the partitioning of the cell material occurs and the complete division of the cell takes place. So you can have mitosis without cytokinesis. That would be nuclei separating, but no cells actually separating. And there are some examples in nature of this. Now, cytokinesis in animal cells and plant cells is different for an animal cell versus a plant cell. In an animal cell, like you see on the left-hand side of the slide, what you begin to see is the cell membrane sort of pinching in and squeezing off to eventually separate into two cells. And hopefully you can get that from the diagram on this slide. On the right-hand side is cytokinesis in a plant cell. Plant cells have cell walls, so what needs to happen in order to have these cells eventually separate from one another is to have a cell wall built between the two nuclei. Vesicles that contain cell wall components begin to migrate towards the center of the cell. Eventually, those components begin to fuse together to form a cell plate, and eventually enough material builds to basically divide the two cells into two cells. It's not unlike taking a single room and putting up a partition in between the two rooms. Now, I always like to add this slide and the next slide after this just to give you a little bit more of a real representation of what's happening during mitosis and cytokinesis. Obviously, in the first slides we just looked at, those are all cartoon versions of cells and their chromosomes. But what you're looking at in this particular slide is a time sequence of actual cells. In this case, though, there are dyes that have been bound to specific portions of the cell. The bluish color is a dye that binds specifically to DNA. The red color binds to actin, which is a cytoskeletal component, and the green color binds to microtubules, again, another cytoskeletal component. So in interphase, what you can see is, again, the chromosomes are not clear, defined structures. It's sort of diffuse nucleus that you see where the DNA is found. If you look at prophase, you can start to see chromosome structures appear, and that's because those chromosomes are, in fact, condensing. There begins to be a little bit of organization of those microtubules, again, in the greenish color. In prometaphase, you can see those chromosomes much more clearly and distinctly. You can also see that the microtubules are attached to those chromosomes. Metaphase, you can see the alignment of those chromosomes along a central axis. Anaphase, the sister chromatids pulling apart towards opposite ends of the cell, again with those microtubules doing the pushing and the pulling. And then telophase, where you can almost see where that separation is going to take place, the chromosomes decondensing in this telophase state. This is also another set of animal cells with different types of stains, but again, you can get a little bit more real representation of what's happening during mitosis in each one of the stages. All right, so time for another concept-checked question. This one hopefully is going to require just a little more thought on your part. So the question is, what would be the result if a chromosome did not have a kinetochore? At this point, go ahead and pause the lecture, think about that, and then you can move ahead and look at the answer on the next slide. So to answer this question, you have to understand chromosome structure and what a kinetochore is and what a kinetochore does. The kinetochore is the specialized part of the centromere. The centromere, again, holds two sister chromatids together, but the kinetochore portion of the centromere is specifically where microtubules attach. If there were no kinetochore on a chromosome, there'd be no attachment point for the microtubules. The microtubules are what push and pull the chromosomes throughout the entire process of mitosis. So the movement of chromosomes to end up along a central plane in metaphase, the pulling apart of sister chromatids in anaphase, that's all dependent upon the movement of those structures, and the way that they move is by microtubules, and the microtubules attached to the kinetochore. So if you don't have a kinetochore on your chromosome, you have no attachment point for the microtubules. That chromosome cannot move in order to get into the proper positions during mitosis. And when we get to meiosis, that same concept will hold true as well. So what we're going to do now is transition into meiosis and reviewing meiosis overall. But before we do that, we need to make sure we review mechanisms of reproduction. These are things, again, you should have heard about in previous courses. Basically, the difference between asexual reproduction and sexual reproduction. With asexual reproduction, you have a mechanism to produce individuals that are not genetically distinct. They are genetically identical to the original individual in question. There are a number of examples in nature of asexual reproduction. The one example you have here is a hydra, where a small bud is beginning to form on the hydra that will eventually fall off and form a new individual. While that forms a new individual, it is genetically identical to the individual from which that bud came from. This is common in the plant kingdom. There are many examples of asexual reproduction there. For example, strawberries. Strawberries are known to form runners from one plant to another. Eventually they break off, and that leads to multiple plants being formed. But if you analyze the genetics would be completely identical to the original quote-unquote parent plant. Sexual reproduction from a biological point of view is going to involve the combination of distinct gametes in order to produce a genetically unique individual. In order for sexual reproduction to occur, again, you have the fusion of distinct gametes. Those gametes are in a haploid state. When they come together, they form an individual single cell that is in the diploid state, and then that individual continues to divide and produce the offspring. So with sexual reproduction, what we have is the fusion of haploid gametes, and the consequence of that is genetic variation, in that you are mixing genetic material to produce distinct individuals genetically. Okay? So the images that you see on the right are an example of what happens from the biological perspective of sexual reproduction. You have gametes that are produced by two individuals in this case, and those gametes come together to produce genetically unique individuals as offspring. What hopefully you will gain as part of your knowledge base as we continue on through the lecture series here this semester is that the sexual reproduction allows for a mixing of genetic material and is one of the key components that allows for genetic variation. So let's give a brief overview of the mechanism of meiosis. Meiosis contains or consists of two consecutive cell division processes, Meiosis I and Meiosis II. In this slide, this is a quick overview of the process. In Meiosis I, you have a diploid cell that undergoes a reduction in the chromosome content, where each cell that results is now haploid. So the original diploid cell has two complete sets of genetic information. The resulting haploid cells at the end of Meiosis I have one complete set of genetic information in them. Meiosis II is another mechanism of division where you continue now from the haploid state to produce haploid cells. All right, so let's go ahead now and review the mechanism of meiosis. You have interphase in a cell that is going to undergo meiosis. Specifically, just like we talked about in mitosis, in the S phase of interphase is when all the genetic material is copied or duplicated. As the cell enters into meiosis, we first enter Meiosis I, which is going to be the reduction in chromosome content phase. Prophase I, you begin to see the chromosomes condense, you begin to see the nuclear envelope break down, you begin to see those microtubules organize. So at this point, it sounds very similar to what we just talked about with prophase in mitosis. However, in prophase I of Meiosis I, there's also an additional component that occurs. Because we are dealing with a diploid cell, we have homologous pairs of chromosomes in those diploid cells. So in prophase I of Meiosis I, those homologous pairs come together in a mechanism known as synapsis. So chromosome number 1 finds its homologous chromosome number 1 and pairs up. Chromosome number 2 finds its homologous chromosome number 2 and pairs up. And this occurs for all of the sets of chromosomes. So using our human example, there would be 23 pairs of chromosomes that have come together in prophase I. In metaphase I of Meiosis I, you see those homologous pairs that have come together align along the central axis. Again, this is all due specifically to microtubules that have attached to the kinetochore portion of the centromere, and that pushes and pulls those chromosomes towards the central axis. In Anaphase 1, the homologous pairs separate from one another and move towards opposite ends of the cell, again with the microtubules pulling them towards opposite ends of the cell. In telophase 1, we begin to see a little bit of chromosome relaxation, but the most important thing is that at the end of telophase 1, you have cytokinesis to form two distinct cells. Each one of those cells will continue on into meiosis II. So at this point, at the end of meiosis I, we have reduced the chromosome content by 1/2. We now have two haploid cells from what started off as a diploid cell. Each one of those resulting cells will continue along in meiosis II. And meiosis II, the mechanisms taking place are just like we talked about with mitosis. Prophase 2, chromosomes condense, the microtubules organize. Metaphase 2, we have the chromosomes aligning along the central axis because the microtubules have attached and are pushing and pulling those chromosomes along the central axis. Anaphase 2, sister chromatids separate and pull apart from one another because of microtubule movement. And then lastly, telophase 2, followed by cytokinesis, will result in the complete separation of those cells. So at the end of meiosis, you have 4 what I refer to as daughter cells that have formed from the one original parent cell. The one original parent cell is diploid. All 4 of the resulting daughter cells are haploid. All right, so this is just a summary to compare mitosis and meiosis. With mitosis, you have a cell that has genetic information that is duplicated or replicated during the S phase of interphase, and then the process of mitosis will begin. What you have at the beginning of mitosis is a single parent cell, and at the end of mitosis, you have 2 resulting daughter cells. Typically, when we talk about mitosis, we talk about the conservation of chromosome content, in that whatever chromosome number you have at the beginning of mitosis is what you will find at the end of mitosis in each of the daughter cells. So, for example, if this were a human cell undergoing mitosis, there would be a total of 46 chromosomes in the parent cell to begin with, and at the end of mitosis, there would be 46 chromosomes in each of the daughter cells. There's a conservation of chromosome content, and the cells that arise are genetically identical to the original parent cell. In meiosis, however, what you have is a little different situation taking place. You start off with a diploid cell as the parent cell. The end result of meiosis, both meiosis I followed by meiosis II, is 4 daughter cells that are haploid. So you start off with a diploid cell, that is the parent cell, and you have 4 haploid cells that result. These cells are not genetically identical to one another, and that is because of the separation of homologous chromosomes that is taking place in this process. And we will come back to meiosis, obviously, when we talk about genetic variation. So it's really important for you to know the stages of mitosis and the stages of meiosis, how they differ from one another, and why one is so important for genetic mechanisms, namely meiosis, because this is how gametes are formed, and this is where we get genetic variation from.